Hello, welcome to SPMM Revision on Demand. In this podcast, I will discuss certain aspects of clinical trials. Let us first consider the flowchart for an RCT. What you see here is called a two-arm parallel RCT, as there are only two treatment arms, active and placebo, and the two arms receive the respective interventions in parallel without any intermixing between the arms. That is, if somebody is destined to receive placebo, they will only receive placebo, and if they are allocated to active treatment arm, they'll only receive active treatment. Random allocation enables the unbiased comparison in this design. So randomization lies at the heart of the RCT. Randomization is often done using a simple randomization procedure. In this scheme, a random sequence is generated using a computer. Let us assume we need to allocate 24 subjects to one of two treatments, A and B. We need 12 people in each arm. The random sequence generated by my computer is pasted here. Suppose if the study has to stop at some point due to lack of resources or any other reason, this sequence of allocation can produce some problems for us. We will only have 5 patients getting treatment B, while 10 would have got treatment A if the trial has to stop at the point shown in this sequence. A meaningful comparison between treatment A and treatment B may not be possible, as these two arms are now unequal. This problem can be avoided if we use block randomization instead of simple randomization. Here, the random allocation occurs within blocks of specific size. We can randomize using blocks of 4, blocks of 6, blocks of 12, etc. Let me show you an example of a 4-unit block here. We're going to allocate 24 people, 12 each, to treatment A and treatment B. If I generate a block randomized sequence as shown here, this ensures that the two arms of the study are equal sized at any given time point of the trial. But still, one problem in block randomization is that you can predict the treatment allocated to the last subject in a block, especially when the blinding is weak. So, block randomization is often used in randomized controlled trials where you want to ensure that if the trial stops in the middle, you will have equal number of subjects allocated in both arms. Now, let us consider some variations in trial designs. Firstly, let us discuss crossover RCT. In a crossover RCT, we initially randomize the study participants to active and placebo groups. In this figure, you can see group A denotes a set of individuals who got allocated to active treatment initially, and B denotes those who got allocated to placebo. In the first stage, you follow them up and evaluate treatment response as you would do in any parallel two arms RCT. In the next stage, group A is given placebo treatment and B is given active intervention. Thus, a crossover takes place with another follow-up stage. This is called a crossover randomized controlled trial. A washout period is often required between the two stages of a crossover RCT. Note that not all treatments can be tested using a crossover design. Importantly, treatments with active ingredients that stay in your body for long periods cannot be tested with this design. For example, depot medications. This design works only for conditions that are controlled by a treatment, but not cued completely by the treatment. If the active treatment would completely eliminate a condition, then there is no point subjecting a group to placebo treatment at stage 2 of the crossover trial. Finally, as you might have noticed, each individual undergoes 2 treatment procedures in this design. So, we can achieve the required power with just half of the sample size. Next, let us consider factorial RCT. This design is very useful if We are not just interested in whether treatments A and B are effective, but if we want to know if the combination or the interaction between A and B will be of any use for the patient. For example, we know that both antidepressants and CBT are effective for panic disorder. We can now design a factorial RCT to see how effective the combination of these two will be in treating patients. In this design, you will have 4 arms. The first box represents the arm where both CBT and antidepressants are given. Second box represents the CBT only group. Third represents the antidepressant only group. And in the fourth box, you see patients who are receiving neither CBT nor antidepressants. In other words, this group may be getting a placebo pill. This is a factorial RCT. Now, let us consider an N of 1 trial. As the name suggests, N stands for sample size. The sample size in this trial is just 1. That is, only one subject undergoes the trial. You can compare 2 different interventions in a single subject. You can do this under double-blind conditions, both clinician and the patient not being aware of the treatment that's being given. In fact, this is the best type of study to answer a specific treatment question for the patient that you see in your clinic. How is this done? In this picture, you can see 3 cycles of a N of 1 trial shown. In a random manner, the patient initially gets either placebo or the active drug. In this figure, I've shown you the placebo treatment occurring initially. Then a washout period is given. This is followed by the active drug treatment. Now this completes 1 cycle. The cycles are then repeated. At the end of every phase of treatment administration within a cycle, the outcome is assessed in a blind fashion. At the end, all results are collated and the clinician and patients can see for themselves which treatment has worked best. This design is useful for choosing between treatments for which there are no clear rationale to make a choice. Let us now see the design for an uncontrolled trial. These are also called before and after trials, and there is only one group that is being studied. Data from this group is collected twice, so the group acts as its own historical control. This design is useful to evaluate the impact of services. For example, you may want to evaluate what difference does a crisis team make in your area of practice with regard to the self-harm rates. Get the historical data for self-harm before the crisis team started operating and compare this with the data obtained from a period after the crisis team started to operate. Please note that this is not a randomized design, so this is prone to a number of systematic errors. Okay, a term that you will often see in the context of RCTs is intention to treat. When analyzing the outcome data from an RCT, you are carrying out an intention to treat analysis. If you follow the principle, once randomized, always analyzed. What does this mean? This illustration from JAMA displays what really happens to subjects who get randomized to a particular arm of an RCT. Some may complete the treatment as planned, some may never take the prescribed treatment, some may discontinue midway, some may die for various reasons, some may stop treatment earlier than advised, some may even end up receiving some of the treatment that they obtained from other sources. Let us call all those who did not complete the treatment as per protocol as dropouts. Imagine a study where 100 enter each arm, but 10 drop out of each arm. At the end of the study, you report the results on the basis of only 80 subjects. Then you are reporting per protocol analysis. This is suitable for early phases of a clinical trial where we are interested in knowing the physiological effects of a drug. If at the end of the study, you report the results on the basis of all 100 subjects who entered the trial, you are reporting on the basis of your initial intention to treat. You initially intended to treat all 100 who got randomized. This is the ideal way of reporting an RCT aimed at demonstrating the clinical effectiveness of a drug. That is, the real-world effect of a prescription that mimics what happens in clinical practice. You might have noticed an obvious paradox in intention to treat analysis. We want to include the outcome data for dropouts, but dropouts are the ones who do not provide us the final outcome data. So how to deal with this? There are various ways of dealing with the missing data. One method is what we call as LOCF, last observation carried forward. Let us assume that there are 7 weekly assessments after the initial observation in a trial. A patient drops out of the study after 3 weeks, then this value is carried forward and assumed to be his or her final score at week 8. If the study has a binary categorical outcome, for example, response versus no response, then we can assume that all of the dropouts in the active treatment group did not respond, and all of those in the placebo group showed a response. This assumption is an extreme case scenario. To be precise, this is called worst case scenario assumption. This is one method of dealing with missing data. We can also do what is called as an imputation. Imputation refers to substitution of a value with another value. We can substitute all missing values with the mean of the values from subjects who completed the protocol. For each missing person, we can also find a person among those who completed the protocol who is very similar in profile, for example, the same age, gender, and baseline score. We can then substitute the missing person's score with this matched person's score. This is called as hot deck imputation. It is a well-known fact that mere participation in a randomized controlled trial increases the response rates for various medical and psychiatric conditions. How can we explain this? Firstly, RCT patients are volunteers. There is something about people who put themselves forward for research that sets them apart from those who do not. They may be more motivated and actively seeking methods of getting better. Also, the strict criteria used for including subjects in many RCTs unfortunately discards patients who have complex needs. For example, in most antidepressant trials, people who have a harmful pattern of alcohol use are excluded. This inflates the response rates in an RCT. It's also well known that sugar pills, placebo pills, have some therapeutic effect on their own accord. This may also inflate the response rates that you see in a randomized controlled trial. Finally, the mere fact that we record outcome in a structured fashion with frequent contacts for RCTs inflates the reported response rates from clinical populations. So, summing up all this, an RCT is far removed from real-world issues that you see in your clinic. This issue is called as the issue of generalizability and is very important. When we conduct an RCT, we draw a sample from the reference population and we aim to apply these results to the target population. The ability to apply the results of a sample to the population is called generalizability or external validity. As discussed earlier, this is limited for an RCT. Even though the internal validity, that is, the degree to which the results are not biased on the basis of the conduct of the study, is very high for an RCT, the external validity is usually poor. So what can we do about this? One way of improving the generalizability is making some changes in the design of an RCT. If we make some common sense changes to reflect real-world clinical practice, we can get a pragmatic RCT with higher degree of generalizability. Some of the changes we can make includes avoiding blinding, providing no extra follow-up, conducting intention to treat analysis, having realistic exclusion and inclusion criteria, employing regular clinical staff and not academic specialists to oversee the treatment plans, etc. These changes will make an RCT more naturalistic. So to recap, we have seen two methods of randomization, simple randomization and block randomization. We discussed the variations in clinical trial designs. Specifically, we saw the designs of crossover RCT, factorial RCT, N-of-1 trial, and before and after trials. We also discussed intention to treat analysis concept and how to deal with missing data. Finally, we discussed the issue of poor generalizability in an RCT and what to do about it. Now, it is time to ask yourself a few questions. Thank you for choosing SPMM course for your revision.